Okay, welcome back. I will introduce David, but first I would like to say that we are very proud and honored to have these international stars here in Lund and in Sweden. It's really fantastic, and to listen to them and to discuss B-cell tolerance, which is fundamental discovery. And they both did these discoveries in independent ways, unknown of each other, in a similar way. The time has matured for this type of experiments that led to a breakthrough. I will tell you a little bit more about David Nemazee. David was actually born in Iran. He moved as a 3-year-old child to US, so basically American, and ended up as a PhD student at Harvard. But then he made an international tour, so he joined to Basel Institute of Immunology in Switzerland, which was a really great institute for European science. And here, for example, Susumu Tunagawa discovered the VDJ recombinations. He got the Nobel Prize for that. So it's a great place, especially for B-cell research. And I think that, but they will say that they picked up some ideas of this here and initiated the work that he's prized for today at the Basel Institute. He continued to make his group in Denver, in US, and then later on being a professor in La Jolla in California. It's really an international tour in his career here. So what has David done? Similar to Chris, he tried to pose the question, are there any B-cell tolerance? And at this time, you could make transgenic mice in doing this. And what David did was to make a mouse that was had B-cells that was specific for MHC class 1. It's one of the MHC antigens on cells. And he crossed that with natural mouse strains because MHC class 1 is polymorphic, so you have different alleles. So you can make an antibody against the self class 1, but you can also have this crossed to something that is foreign because it's an allele. So when they crossed that, they saw B-cell deletion on here. And so this was in parallel at the same time. But what he also did, and it was published in Nature in 1989, he also discovered was something new. So in parallel with Martin Weigert, they discovered also a new mechanism that was really a surprise mechanism how this could work. That when the B cells got deleted, it's kind of a waste of the production of B cells in the bone marrow, of course. But then they discovered that these B cells that have self-reactive B cell receptors could be rescued by making another recombination. And they call it receptor editing. So then you can rescue these B cells and they can do something, good job anyway, right? So he discovered a mechanism of receptor editing as well. So I think I will give the word to David now and listen to your talk.
Thank you. Okay. Thank you very much. I want to start by many thanks to the Academy, but especially to the donors, the Crafoord family. It's breathtaking, the generosity, and I really appreciate it. So I'm going to basically talk about, first a little bit about B cells. You've heard a lot, but just my way of looking at them. So this, for someone who isn't an immunologist, the way I think about them is if you have a B cell, they're all different. You see every one has a unique receptor, and if one sees a virus, it can proliferate with CD4 T cell help. And it has this receptor that can bind the virus, and that goes along with proliferation. That receptor is increased in number, and it can change flavor, for example, from an IgM form to IgG, and this eventually this receptor gets secreted as antibodies, and you also make these memory cells that can have somatic mutation that usually improve affinity for the virus or what have you. Chris talked before about Paul Ehrlich, and who introduced this idea that there must be a mechanism to promote tolerance or to avoid autoreactivity, and it was all based on a very simple experiment where they could immunize or inject red blood cells from one goat and into another goat, goat B. And what they found was that the goat B could make antibodies specifically to that goat A that could not only bind to the cells but destroy them with fresh serum. And so it was obvious that any goat has a potential to make autoantibodies or to destroy itself. And that concept is horror autotoxicus. And in fact, if you look in the human population, it's estimated that several % of us actually make some antibodies or have a T cell response that is just destructive. And so, as Chris mentioned, you know, there was a huge literature that suggested or theorized that there is a kind of tolerance to avoid autoantibody production, and it involves weeding out from among these many different kinds of B cells those that are autoreactive. And by this way of thinking, the tolerance occurs through a cell death mechanism and is usually thought of as part of the clonal selection hypothesis. This we all accepted, really, to some extent, but the mechanism wasn't really well known. Chris and I are receiving this award for discoveries in B cell tolerance, but when we joined the field, it was already a field with a lot of very smart people. Chris mentioned Gus Nossel, but at Scripps we had two other men who worked on B cell tolerance, William Weigel and Norman Kleinman. And actually Norman was the person who recruited me to go to Scripps. So this was a field that had been worked on very hard for many years, and as Chris mentioned, was still not fully accepted by many other scientists in the field. And so to illustrate the problem, in those days, to investigate one of these rare B cells, each with its a rare specificity, you could only measure that cell by virtue of its making an antibody. So you could, let's say that's cell number 9, you could activate somehow and measure its antibody, its corresponding antibody, but without that it was really difficult to study. And how could you do it? And in fact, it was quite difficult. So here are a couple pictures from papers of Gus Nossel and Norman Kleinman and their collaborators, where they actually carried out experiments to induce tolerance and measure the effect. And these were heroic experiments. So in the case of Nossel and Pike, they had to deposit individual B cells in a well, in a tissue culture well, and then measure the antibody production, comparing tolerized or untolerized cultures. Or in the case of the Norman Kleinman experiment, they were measuring mice that were tolerized at different ages, and it was clear from Norman's work that young mice were easier to tolerize than older mice. And again, he had to generate a very elaborate culture system to measure these rare antigen-specific cells. And these papers were dated 1975, 1976. And so you should appreciate that at the very same time that those experiments were being published, there were counterexamples. This one actually from Stockholm. So other researchers who were very credible, working at the very same time, came to opposite conclusions. And this is, Chris sort of touched on this. So here's an experiment where the investigators compared mice who were tolerized or not tolerized, then just stimulated the cells with lipopolysaccharide to get them a large number to secrete antibody. And here you see these bars here show roughly the same amount of specific antibody secreted. So in other words, they concluded there was no tolerance. And you could understand that it was hard as a student starting out in the field to understand what was going on or how anyone knew anything, and how could you sort this kind of question out. And so what we really wanted to do was to simplify the system so that you could be sure of the answer you were getting. And to address that, you had to address the diversity of B cell specificities. So why do you have all this variety? How does that arise, and what can you do about it? And so in explaining this, I'll be explaining why you turn to transgenic mice and how that helps you. And it helps you to understand how we understood editing later. So the first thing you should know is there is an asymmetry in the antibody genes that helps explain part of it. So, antibody genes are generated by two sets of loci. There's a heavy chain, antibody heavy chain locus that has V, D, and J elements. And those those gene segments get combined to make a finished gene, and that's why in individual B cells this rearrangement happens independently. So that's how every B cell can have a unique receptor. So that V, D, and J elements are assembled on the heavy chain to give rise to the heavy chain protein. And on the light chain loci, you have V elements and J elements that get recombined that eventually give rise to the light chain. And these get assembled into the antigen receptor, the antibody on the B cell surface. And this is how you generate this amazing diversity in B cells. But I just showed you that there was this asymmetry in that the light chain loci don't have these D elements and the heavy chains do. And there's a second asymmetry in the order in which these genes are rearranged. So first, in B cell development, you rearrange the heavy chain genes, and then somehow the B cell knows it's made a good heavy chain rearrangement. 2/3 of the time they're not functional, so it has to try on the other allele. But B cells never express two functionally rearranged heavy chain genes. they can sense the presence of a good one. And that was known way back when we started our work. And then when a cell with a good heavy chain then goes on to make a light chain, and these are assembled to give rise to IgM-positive cells that later in development co-express IgM and IgD. So around the time we started our work in Basel, these papers were coming out from Storb and Brinster, Rusconi and Kohler. They basically showed that when you introduce into the germline of mice an already rearranged gene for antibody, light or heavy, or both, that suppresses endogenous rearrangements. In other words, you can subvert this feedback regulation system of normal B cell development to block further endogenous gene rearrangements. So if you introduce a transgene that encodes an immunoglobulin heavy and light combination, that tends to suppress the endogenous genes. So my contribution to this field basically was to choose an antibody, which we chose the antibody 383. And it's a kind of an antibody like that goat anti-A antibody. It's an antibody that sees a cell surface protein that's present in some strains of mice, but not others. And after choosing the antibody we wanted to study, I had to clone these genes. I had to first learn how to do it, and from friends and colleagues, and then to do it. And then I needed help to make a transgenic mouse. And fortunately, in Basel, there was this young embryologist, Kurt Bürki, who knew how to do the microinjections and helped us make these transgenic mice we wanted to make. And so the concept was to, if you gave appropriate heavy and light transgenes that were already rearranged, you introduce it into the germline of a mouse, you can make a mouse where every cell is identical. And even someone like me could figure out, without single cell culturing or anything fancy, you could just look and see what happened. The mouse would do the experiment for you on the right MHC background, on the right genetic background. The antigen would be there. On a different genetic background, the cognate antigen would not be there, and you could compare the mouse strains. So I should give a shout out to my friends and coworkers in Basel. So when I was in Basel, I had a small lab and one technician and a budget. And that was the system they had there for junior scientists. So I worked initially with one technician, first with Suzanne Studer, and then Chantal Guillet, who's pictured there on the bottom left. And I had to learn a lot of molecular biology, so I teamed up with some postdocs in Michael Steinmetz's lab, who basically, out of the goodness of their hearts, helped me to learn some of the technology. And these are my friends Mike Kiefer and Zlatko Dembic. Later on, Zlatko, who was an expert in Cosmid library production and screening, who was very instrumental in cloning T-cell receptor genes in Steinmetz's lab. They helped me a lot. And then I had other friends. And some of these fellows, like Andrei Traunecker, was actually Susumu Tanagawa's technician, one of his technicians, and he was a fount of knowledge that helped me a lot. And I had a lot of other friends who gave me pointers from time to time. So that was critical when I started out, because I really didn't know much of anything. So ultimately, we were able to make mice that we desired. So this was the best mouse we made. We made several versions of these. And so on the top is just a non-transgenic mouse, and we're staining cells on the horizontal axis. That's cells that carry IgM, which is virtually all B cells in a mouse. And on the vertical axis, that's the cells were stained for the idiotype. So what the idiotype is just the variable region structure on the antibody. So I had to raise an antibody to that 383 antibody, and that's the idiotype. And so that allowed us to track the transgene encoded antibody, or B cell. So in the best situation, virtually every cell in this mouse is what we programmed in the transgene. And so that sort of made things easy to study. You can just look at those cells and see, are they there or are they gone? So this is a mouse lacking any autoantigen. And then we did a series of different experiments. I'm showing you a little bit out of the historical order in which we did these experiments because it makes it a little easier to understand. So one kind of experiment we did was to breed our transgenic mouse to a mouse in which the antigen that the B cells saw was exclusively expressed on the surface of liver cells. And this was another transgene we obtained from Jacques Miller's group, who's studying T-cell tolerance to that same class 1 MHC molecule. And then we could ask what was the fate of these B cells. And it was very clear. So this experiment, the first author was David Russell. He was my technician in Denver. And what you see, if you just focus on the bottom there, so in the bone marrow, on the bottom left, you see all of the B cells in the bone marrow carried this transgene specificity. And if you look in the spleen, the cells disappear compared to the control, which is a center set of panels where no antigen is present, where we had 39% of the cells expressing the idiotype. And then if you look further to the right at the lymph node population, if you look in the bottom right, the self-reactive B cells are essentially gone. So what we saw was that the autoreactive B cells were deleted, and this result was so straightforward. We had no problem publishing this result at all. It was amazing, and as long as you have a very clean system, people believe you. And so this was sort of clear, but you can see already that we sort of had a mixed answer in the spleen. There were some cells there still. They didn't die instantly. They were hanging around, but they took some time to die to find the antigen, bind it, and then get deleted. But in this system, there were almost no other cells around, and so the answer was clear. So this fit with the original Burnett idea of clonal selection. The autoreactive cells were made in the bone marrow, they go out into the periphery, and if they're autoreactive, they get eliminated. Everyone was happy. But when we look more closely at our central deleter mice, we came to slightly different conclusions. So here, the antigen is present on the whole body. You breed this strain, and you get something similar looking. So this is a slightly different way of showing the data. But if you look in the top right panels, you could see there are a lot of B cells in the mouse lacking antigen, and all of them expressed the idiotype on the surface. But if you look in the top left two panels, in the presence of the autoantigen, no idiotype positive cells were detected in the spleen. But there were a lot of idiotype negative cells there that we couldn't really explain. We assumed it was just leakiness that was happening there. And in the bone marrow, you could see the B cells started to express the idiotype, but later disappeared, or we thought were deleted. And that was sort of our initial take, and that there was deletion of autoreactive cells in the bone marrow. But the better the mouse that we made was, the clearer, the easier it was to find anomalies. And this was the interesting anomaly. So here we stain the spleen cells of this central deleting mouse. So here all the antigen is everywhere. All the B cells are reactive to it, and you delete the major clone. But here we stain the B cells with an antibody to the heavy chain that was the transgene encoded heavy chain. And on the bottom, we're using antibodies to the lambda light chain. So our particular transgenes used a kappa light chain, so we knew anything that was lambda must have been encoded by the endogenous light chain genes. And so there was a clear population that had aspects of both. There was the transgenic heavy chain, but a different light chain. And this led us to hypothesize that in the bone marrow, as B cells develop, they first make a heavy chain, and then they rearrange and express the light chain. So when this is innocuous, that can turn off further rearrangement. But when the B cell encounters self-antigen in the bone marrow, rearrangement isn't turned off, recombinase continues, and that promotes light chain rearrangement and changing of the light chain specificity. And so really we started our studies thinking we were going to prove the clonal selection hypothesis, but in essence we were testing it, and we found that it didn't really fit the facts, and that a lot of the initially autoreactive cells were not deleted but could change their specificity. And this is why we came up with the term receptor editing or receptor selection. So it's really a different concept than clonal selection. And what somehow made it seem more believable was it helped to explain the asymmetry in the immunoglobulin genes. Remember I told you that the heavy chain genes have these D elements that rearrange, and the light chains don't. So when the heavy chains rearrange together, they consume the D elements and can't rearrange further because there are upstream V's and downstream J's, but they can't rearrange to each other. Whereas on the kappa light chain locus, and in human lambda as well, there are no D elements. So if a V, for example, V4 rearranges to J, you can get leapfrogging rearrangement that could delete the first light chain and replace it with another very efficiently. And we were lucky to figure this out, because in the conventional transgenics, this doesn't happen efficiently. We were just fortunate that we could see it, and that lambda was so predominant in this mouse. And if you run out of J elements on the kappa locus, you can get rid of that even, because there's another element called the kappa deleting element that can inactivate the entire kappa locus, and this kind of rearrangement occurs in virtually all cells that go on to make a lambda light chain, which is a lot in humans, about half our B cells. So our view of the system is that initially we thought that apoptosis was a major mechanism of central tolerance. Well, now we think it's very minor, and that most central tolerance in the bone marrow goes through a receptor editing pathway, and then at a certain stage when the B cells are have matured enough and go to the periphery, they can't edit very efficiently, if at all, anymore. And the tolerance mechanisms are quite different, as Chris described. So we think early on receptor selection is dominant, and later on clonal selection mechanisms like what we saw in our liver-specific autoantigen mouse. Now I should say I had a lot of wonderful colleagues in doing this work. In Basel, it was mainly Kurt Berkey and my friends who helped me out a little bit. But when I had my own lab in Denver, I had a lot of very fantastic people who did a lot of important work that extended the studies. So, for example, Doron Melamed showed that editing could work in vitro culture systems that helped people believe us a bit more. Mark Redder looked in normal B cells to find evidence for editing and tried to estimate how frequently it happens in the normal immune system, and actually was the first to show it probably happens a lot. I mentioned David Russell. Mike Chumley asked, what happens if you start with a B1 type B cell receptor? Does that induce editing in bone marrow cells? And his results are too complicated to explain, but it was interesting. Yeah, so we had a number of other useful contributions there. Actually, Jenny Kench on the right did an interesting experiment. She bred our transgenic specificity to a mouse strain that gets lupus, and asked, do you still see peripheral tolerance on the lupus background? And what she found was very interesting, that you still delete this major clone in lupus-prone mice, but they still make autoantibodies to DNA and other things. So it was interesting you could get tolerance to certain autoantigens, but not others on that genetic background. And then when I moved to La Jolla, we got into other kinds of studies. A lot of studies on BAF and that pathway were done by Amanda Gavin. And Jose Vela knocked out the kappa deleting element locus and studied those mice. Laurent Verkozy studied RAG regulation and so on. So a number of really wonderful colleagues that we worked with, and I'd like to thank them. They're quite responsible, and thank you again for this wonderful prize and inviting me here. Thank you.